This is Kyle Bell with the South Bend Voice. Today we are speaking to Matt Eng and Sean Ryan, both of whom are PhD candidates in biological sciences at the University of Notre Dame. Sean is the founder of the Pieris project. Both Matt and Sean are part of a crowdfunding project that hopes to study the effects of climate change on biology. Thanks for joining us.
Thanks for having us, Kyle.
Thanks.
Matt and Sean, first could both of you briefly tell us about yourself?
Yeah, so I am a PhD candidate at University of Notre Dame, and so I'm in my fourth year here, and I actually work on mosquitoes. And so I know the Pieris project works on butterflies, so it's a little bit different, but I do have some familiarity with entomology. And so I just know Sean through the department and whatnot, but I'm a big fan of actually engaging the public within scientific research, and so I was interested in joining Pieris project for that reason.
Yep, and Sean Ryan here. I'm also a PhD candidate in Jessica Hellman's lab. Most of my work is looking at the impacts of climate change on insect populations, and I do study butterflies for my research, looking at a butterfly hybrid zone and looking at how climate change may be affecting hybridization between the two species. But as Matt was saying, I'm also very interested in citizen science, and for a while now I've been interested in engaging the public in scientific research because I think there's just an amazing amount of things that you can do when you open that door in terms of both improving the science and then also sharing what you're learning with the public.
Can you tell us more about the Pieris project? As I understand it, the goal is to see how the environment affects butterfly species, which is known as the cabbage white?
Yep, that's correct. So about 150 years ago, the cabbage white butterfly invaded through Canada and northeast U.S. all the way across, within about 20 years, all the way across to California. So it's pretty amazing then that within the last 150 years, this butterfly has come to conquer basically North America. But then also within the last 150 years, it's also invaded Japan, New Zealand, Australia, and many other parts of the world, and that's because it eats cabbage, hence the name cabbage white, and many other plants in the mustard family, so kale, Brussels sprouts, mustard. So it eats the things that we eat, so it's no surprise that as we started cultivating those plants, it was easy for this butterfly to invade. And so during that 150 years, as the butterfly spread into these new environments, its genome, or the genes inside of the butterfly, have likely adapted to the new environments that it now encounters and now inhabits. And so we want to collect butterflies from all across the range to see what has changed in terms of the butterfly's genome and also in terms of its phenotype, so its color and its shape. We have one student who's in University of Nevada, Reno, Ann Esposette, is looking at how agricultural use or practices, so the amount of nitrogen in their food influences the color of the butterfly. So there's a number of things we can do with a collection of butterflies from across the world, but mostly looking at how the environment that it lives in now has influenced its genes and its phenotype, or how it looks. And then we can also reconstruct its invasion history. So we can look at, where did this butterfly come from? We actually don't know. We know it's somewhere in Europe, but we don't know the exact location. And it may have invaded multiple times. So it may have come over multiple different times, and actually from different countries as the origin.
So how many continents is this butterfly in right now? You said Europe, North America, and Asia?
Yep. North Africa. Yes, it's pretty much everywhere except for Antarctica.
Okay.
So it may well be the most widespread and abundant butterfly on the planet to date,
okay,
but only as of the last 150 years.
And how can studying butterflies tell us about the ways that other species might respond to climate change?
Insects are good indicator species because they're so-called cold-blooded. So their biology, their metabolism is directly related to temperatures. So they're usually one of the first organisms to respond to changes in their environment, especially climate. But evolution, there's also much we can learn in terms of general biology or evolution of this organism by looking at how. So I should say that invasive species present a novel opportunity, or a great opportunity to study how species adapt to changes, because they're kind of a natural experiment. They've recently invaded many different locations, and so they're presented with new environments or new conditions that they have to respond to and potentially adapt to. And so they're a great system to explore questions about how organisms adapt to changes in their environment. And we can explore those in the cabbage white. And some of what we find could be applicable to other organisms.
And you mentioned that you've been studying butterflies. What other studies have you done?
Me personally?
Yes.
So as an undergrad, I looked at fire ecology in plants in California and a maritime chaparral environment, so basically coastal shrub communities, and looking at how soil moisture, so the moisture prior to fire. So if you're not aware, a lot of times for management purposes, there's prescribed fires, so burning of the landscape in California and places to keep the communities or to manage the plant communities. And so I was looking at the effects of moisture, so like maybe previous rain, how that might be influencing the plants' germination, so how they respond to fire during a wet season or rainy season. But I've also looked at, for my master's, looking at how the genes within plants influence the chemistry of those plants and then in turn influence the preferences of insects. So we could work with Arabidopsis thaliana, which is a model organism for people that work with plants. It's an amazing system because you can basically buy plants where we've engineered the genes that influence the defense chemistry, so the chemicals that help them defend themselves against herbivores. We can buy plants where we've altered that chemistry, the genes that alter those chemistry, and then look at how that affects the herbivores that feed on it.
And then my expertise is in mosquitoes, and so I work on a very molecular background, and so working on this project is a little bit new for me, but I can say that I know I work with a mosquito called Aedes aegypti, which carries dengue virus, which is similar to West Nile.
Mm-hmm.
So most people in the U.S. haven't heard of dengue because it's tropical. And so this mosquito is something that probably originated from Egypt somewhere or northern Africa, and over the past 150 to 200 years has spread sort of across the globe. And so now, whereas you used to only be able to find this mosquito in Southeast Asia and parts of Africa, it's now in South America, all over Mexico, and it's increasingly sort of increasing its range to include the southern tip of Florida, Key West, and the southern part of Texas. And so this is certainly an insect also that we could potentially study as an insect that has important implications for global health.
So what is your hypothesis going into the experiment? Do you think that the butterflies will be able to adapt to the changing environment, and how?
Yeah, so that's a very good question. So our hypothesis is that there is quite a bit of local adaptation. So that's actually one of the original questions that was asked. I don't know how many years ago, it was a few decades ago, looking at, is there genetic structure? So that is, are there populations that are kind of becoming different and adapting to the environment across the range? And they looked at that in the Northeast, and they only used a few genetic markers, so they were called allozymes, to look for this structure. And they didn't find evidence of much structure. But part of that is because this butterfly's populations are so large that without a lot of genetic markers, it's hard to really find structuring in the population or differentiation between the different population that may be occurring. So one of the approaches that we're taking here is we're using the advances in sequencing and the reduction in cost that's gone on over the last decade to generate thousands of genetic markers within each butterfly that we can then look at. And because their populations are so large, they're actually more likely to be sensitive to natural selection. And so we're likely to find evidence of evolution and even likely to find adaptation. So one of the things is the butterfly now lives down in Florida. It lives all the way up into Alaska and across that range. And the climate or the length of the growing season is very different across that region. So if you look at many species that live at such a large gradient, you find that they adapt to the environment in kind of a latitudinal climb, because that's how climate generally changes as the season gets shorter as you move further north or higher in latitude. And so we're likely to see that as this butterfly spread across the U.S. 150 years ago, now that the dust is kind of settled and they've been adapting over the last 150 years, or I should say evolving over the last 150 years, it's likely that they've adapted to those conditions. At least differences in growing season, but probably other things as well.
I'm sure that some of our listeners will hear about this project and think to themselves, why should I care about butterflies or any other insects for that matter? What would you say to them?
Well, I'm a bug guy. I'm a bug geek. I find that hard to understand. I've fallen in love after taking an entomology course where I think it's hard to appreciate organisms until you really start to get to know them, put them under a microscope, or spend a little time observing them and watching them. I think that's true for any organism. The more you know about it, I think the more appreciation you have, and that's what's great about programs like Planet Earth and things like that. But I would say why? Well, insects are pretty much the most dominant organism on the planet, so they're everywhere, and they're in larger numbers than almost everything else. And they're really, really important for ecosystem function. So, there was a talk on Science Friday here at Notre Dame just the other week about forensic entomology and about flies that you can find on dead bodies to determine, when was the person murdered and the time of death and things like that. But that's what, one way of looking at, how we can use those flies. But those flies are also converting all of these, dead animals back into nutrients into the ecosystems they live in. So that's the ecosystem service that they're performing for us that we don't even ask for them to do, or ants digging holes in the ground and helping water percolate through the ground and things like that. Bumblebees, we're all, I think, aware that, bees are very important for, most of the foods that we eat. So many of these insects are providing services that we're either not appreciating or in some cases not even aware that they're providing.
Yeah, I'll also say that before joining the project, I really didn't have much of an interest in butterflies. I didn't really know what the cabbage white looked like. And then after sort of like a week or two of being on the project and learning about the butterfly, I came to more appreciate how widespread it is. And I began to notice, too, how often I was actually seeing it, but just not noticing that it was there, that it was actually a cabbage white, that it's an invasive species, and whatnot. So I think just learning and being aware of the science that's going on sort of helps the public appreciate nature and some of the creatures that they do come in contact with, but don't necessarily recognize on a daily basis.
So from what I understand of the Pieris project, you're going to have people from across the country and indeed across the world collect this specific species of butterfly, and they will send it to you guys to collect the DNA. Is that correct?
Yep.
Could you talk a little bit more about that process?
Yeah. So when we get the butterflies, we'll actually photograph their wings and then use one of the wings. So Anne, as I was saying earlier, Anne Espesette at University of Nevada, Reno, is interested in the wing chemistry, the coloration. So there's a pigment in their wings that is causing them to be that bright white that you see. Mm-hmm. And she's interested in how the nitrogen in their food, the amount of nitrogen that they're consuming, is influencing that pigment. So she'll be doing chemical analyses on the wings so we can see how that varies across the landscape and whether if they're in more agricultural rich areas, whether they have more white pigment versus other more natural areas. Then we're going to be photographing, and we can look at all kinds of things like size of the wings, the size of the butterfly, and other morphological traits. And then also preserving that information that we have forever. So we're taking a snapshot of what these butterflies look like at a moment in time now, and then doing this again next year and the following year. And having these long-term pictures, these snapshots, are really important for looking at changes through time that you can't get unless you're collecting them each year, collecting a few. And then so the other thing we're looking at is the genetic information. So we extract the DNA out of the butterflies by basically crushing up some of their legs and putting a bunch of chemicals in a tube. And then what we can do now, I don't know how interested you are in the details, but what we can do now is we can basically put little barcodes, attach them to each individual so that we can pull them together and sequence many butterflies at once, which reduces the cost and makes it possible that we can sequence hundreds of the butterflies that people sent in. And then that sequencing, it'll generate tens of thousands of genetic markers from across the genome of each of the butterflies.
Why did you guys decide to go with a crowdfunding approach instead of a more traditional avenue such as a research grant?
Well, the research grants take, 1 to 2 years, perhaps. So you have to write now with NSF would probably be the most appropriate for our research. NSF is more basic research, and the NIH is more human health-related research. So they have what's called a pre-proposal. So you wrote a short proposal of your project, and then this is, well, I'll get to that in a second, but that takes 4 to 6 months, and then you get approved to then write a full proposal. Then you have to wait another period of time, and that can end up taking a year or two to even get approved and get the funding. And since we're doing citizen science, we really want a quick turnaround time, turning those wonderful collections and all the help that we got from the public into data so we can share results, because we don't want it to be like those cereal box prizes that you got when you were a kid and you get excited, you fill out the form, you send it in, and then 3 years later you get the prize. But actually, have a fairly good turnaround time on getting results to show people these are the cool things that we're doing with your help.
There's also national grants tend to award large amounts of money, and so for something like this, we don't need tens of thousands of dollars or hundreds of thousands of dollars. We can do it with just a couple thousand, and so we thought that crowdfunding would be a quicker way to be able to reach that funding goal.
All right. Well, thanks again, guys, for taking the time to speak with us today.
Yeah, thanks, Kyle.
Yeah, thank you.
You can find out more about the Pieris Project at pierisproject.org. That is PIERIS project.org. You can also find out more about it at southbendvoice.com. Search for Pieris Project. Please be sure to like our Facebook page at Facebook